Hello everyone. Today we are going to continue the chapter number 5, Molecular Basis of Inheritance. And in today's class, we are going to discuss mainly the NCERT session 5.2.2, which in this we are going to study the properties of a genetic material, that is DNA, and what we are going to focus on, that what is like difference between, we will compare and contrast RNA and DNA, and from that we will get to know that why DNA act as the genetic material but not the RNA. So let's get started. Disclaimer. So what is going to be today's learning objectives? So today what we're going to see in our slides. So first we will explain that why DNA is the primary genetic material. Why DNA act as the genetic material. Then we will understand that what is the structural benefit, what is the structural advantages of DNA in terms of stability and why it is act as a primary genetic material. Then we will recognize the genetic roles and functions of DNA and RNA, and from here we will get to know that why RNA is not acting as the genetic material whereas the DNA is doing so. So now the index. So in this today's class we are just going to focus on one session of the NCERT, that is 5.2.2, which is property of genetic material, specifically like DNA versus RNA. As I said, we will compare and contrast both the nucleic acid, that is DNA, and RNA. Then at last we are going to give up the summary, give up this sum total of all the slides what we are going to study today. Then there will be some NCERT based questions, some keynotes, and last interesting questions which you guys gonna solve yourself. And after this whole slide lecture, we're going to solve DPP questions that we always do. So now let's start the topic. So first is like what is RNA, like DNA or RNA? Who is the genetic material and why it is so? What was the historical evidence that proved that DNA is the acting as genetic material? So in last class we have studied about this Hershey-Chase experiment, and that was the experiment that proved DNA act as a genetic material. If you guys remember what we have studied in the last class, that how Hershey and Chase has performed that bacteriophage, that blender experiment, okay, in which what they did, if you guys remember, I will give a quick recap. So what they did, they used, what was their test subject? So their test subject was bacteriophage and bacteria. Bacteriophage, we all know what is bacteriophage. Bacteriophage is the virus that infects the bacteria. So their first batch of their bacteriophage is what? It is radio labeled. It is phosphorus radio labeled, and because it is phosphorus radio labeled, that radioactivity is present in the DNA. Whereas if you see the second batch, what it was, the sulfur was radio labeled, means the protein was radio labeled. And what they did, they infected that bacteriophage to bacteria. Then they what did, their next step was the blending, in which what happens, the viral protein coats which was on the surface of bacteria got removed. And at last what they did, they centrifuge it. Why they centrifuge it? So that they can separate this viral particles and the bacterial cells. So now what happened when they centrifuge, because bacterial cell has more density, it came at the bottom of the test tube and it was pellet, whereas the viral particle was in supernatant. So what the result shows, what they showed, that approx 85% of 85% of the radio labeled phosphorus was present in the bacterial cell, whereas the 80% of the radio labeled sulfur was present in the supernatant. So it shows that, okay, so it was the DNA that went inside into these bacteria and it is act as a genetic material. So this was the Hershey-Chase experiment. It was very important unequivocal experiment, as NCERT said, that proved DNA act as what? the DNA act as genetic material. So now, what was the challenge of what was the challenge that uh, to this experiment? So that the thing was that some genetic material, some viruses. Okay, some viruses genetic material is RNA, and that was a challenge of this discovery, that they can say, okay, so this some of the viruses have this RNA as genetic material. So how you guys can say that, okay, DNA is all only acting as genetic material? So this was the biggest challenge. Example of the viruses that contain RNA as their genetic material are the tobacco mosaic virus and QB bacteriophage. So this is the picture of Q beta virus and tobacco mosaic virus. If you guys see, so what it is showing that, okay, this this bacteriophage has what? It has single stranded RNA. This is maturation protein and it is caspid protein. What caspid proteins do, as I told in yesterday's class, it is used to protect the genetic material. Whereas in this tobacco mosaic viruses, it is capsid, the protein which is protecting the genetic material, which is also here what? RNA. So some viruses have RNA as genetic material, and that was the challenge that that can question this experiment. Okay, you are saying that DNA acting as a genetic material, but some of these viruses have RNA as their genetic material. So then, why this? Now the question arises that why this DNA is the prominent genetic material? Why DNA emerged as the predominant genetic material in most of the organism? Why most of the organism, when has seen, we have examined, they seen that DNA is acting as their genetic material, whereas RNA primarily serves dynamic function. RNA do present there and have several functions, but it is not acting as genetic material in most of the organisms except for viruses. So the role that RNA is playing, first, it is acting as a messenger RNA, mRNA. Okay, then it is acting as a tRNA. What is tRNA? It is transfer RNA. Okay, this is act as an adapter molecule, and it is basically, yeah. So they play very crucial role in what? In translating the genetic code from mRNA into protein, and it acts as a kind of intermediate. And okay, and it's one of the more of its function is that it carries amino acids to this. It brings amino acid to the ribosome, and we know that what is ribosome? Ribosome is the site of protein synthesis. So it can act as a mRNA. Okay, we know that first DNA convert into mRNA. Then second, I told that it acts as a adapter, which is the transfer RNA, and it also play very important role in protein synthesis. So this was the role of RNA in most of the organism, whereas DNA is only acting as genetic material. Then, so what should be the characteristic of the genetic material? So a molecule must fulfill the following criteria to act as a genetic material. So what are those criteria? First, replication, means it should generate its replica. Second, stability, that this genetic material should be chemically and structurally stable. Then mutation, it should provide scope for slow changes. Mutation required for evolution, means with time they show mutation, but very slow mutation, and so that evolution can take place. Then expression, it should be able to express itself in the form of Mendelian characteristics. So we know that what was Mendelian characteristic, like how this from parents the progeny can show some traits. So it should express what? It should express some Mendelian characters. Okay, so we know that there are few characteristics, okay, that our genetic material should follow. It should be chemically stable, it should, structurally stable, it should do replication, it should show the Mendelian characteristic and all those things. So now it's time to compare DNA and RNA. because we are saying that DNA is present as a predominantly present as a genetic material in most of the organism, whereas RNA is not. RNA is doing what? It is acting as messenger RNA, it is acting as transfer RNA, it is helping in protein synthesis, doing everything, but it is not the genetic material. Okay, it is not transferring information from parents to progeny, to its offspring. So why? So there are some comparison which will show you that why DNA is acting as the genetic material and not the RNA. So let's see. First option is, first property is replication, that is base pairing and duplication. First, they should be able to do base pairing, that purines can bind it to complement to the pyrimidines. Adenine and guanine can bind to thymine and cytosine. Okay, and second, duplication, that it should duplicate also. So let's see what is like criteria here for both DNA and RNA. So both DNA and RNA can self-replicate due to base pairing rules. Protein cannot self-replicate, making them unsuitable as genetic material. So it was a fight. Okay, so what are the so we have said that 3-4 properties are there that a genetic material should follow. Okay, now we have compared those things. So first property was replication, where we have seen that DNA and RNA both can self-replicate due to base pairing rules. We know that, except the fact that in the case of pyrimidines, DNA has what? DNA has thymine and RNA has uracil, except that they can do base pairing and they can self-replicate. But proteins cannot self-replicate, making them unsuitable as genetic material. So they are out of the race for the first property only. They are out of the race at first only. They haven't fulfilled the first criteria only. So from here we can say, okay, so only DNA and RNA is running in the race. And now see what are the others, comparison and other property they are following. So first property, that replication, both DNA and RNA is following. Conclusion, both DNA and RNA are capable of replication. Now, first was what? Replication. Now second comes what? Second comes DNA stability. So when we talk about DNA stability, let's see who are more stable and all those things. So first, let's see DNA stability. DNA is more stable throughout organism's life cycle. Means DNA is very stable throughout the organism life cycle. So whatever the lifespan of an organism is, DNA will remain stable. It can survive heat. Separate strands can reconnect under proper conditions. Means if their strands even are separated from each other, they can reconnect if given proper conditions. Less chemically reactive, means they are not very reactive, they are chemically less reactive. Thymine provides additional stability compared to uracil. We know when it's about base pairing thing, so DNA has thymine irrespective of uracil. Okay, and what thymine is doing? Thymine is giving more additionally stability when compared to uracil. Preferred for long-term genetic information storage. So here what we have seen, if you talk about the stability of DNA is more stable and it remains stable throughout the lifespan of an organism. It can survive heat also. If their strands are reconnect, that we have seen in this PCR thing, that when we have denatured it, but when we have given it the right temperature, it again, when we have given it primer and right temperature and all those things, it can reconnect also. So it can reconnect or it can duplicate. In the PCR thing, it replicated it with the help of primer and DNTPs. So even if they are separated, given the proper conditions, they can reconnect or they can replicate itself. Okay, it is less reactive chemically. Its chemical structure is such that it is less reactive as compared to others. Thymine is providing its what? It's providing its more stability when compared to the uracil. And what is thymine and uracil? So these are the pyrimidines, and we know that purines and pyrimidines together complement each other, that base pairing happens. But in the case of DNA, it is thymine, and in case of RNA, it is uracil. And thymine is providing what? Thymine is providing additional stability, and it is preferred for what? Long term genetic information storage. So these are the points of the DNA stability. So this is like they have shown a pictorial representation of RNA versus DNA that we will see when we will see the stability of RNA also. So now, if we talk about RNA stability, we have talked about whom? We have talked about DNA stability. If I will talk about RNA stability, it is less stable. Why? Because we know that the ribose, okay, that sugar that present in the RNA, okay, it is ribose. Whereas it in DNA it was what? It was deoxyribose. Means at the 2' position of that ring structure, OH group was absent. But in RNA it is what? In RNA that 2' OH is present and it is reactive. Means it can react. Okay, it can more easily degradable, it can be degraded very easily, it is catalytic in nature, and it is more reactive chemically. And that's the reason that DNA is more stable than RNA. Now you guys understand why? Why? Because this RNA has a 2' OH group presence on its sugar, the ribose sugar, whereas DNA is named so DNA because it was like said deoxyribose, means that the 2' OH, there is no presence of OH group, and that's the reason it is less reactive. But RNA is more reactive and less stable and can be easily degradable, and it is catalytic in nature. And that's why what conclusion we made? We made the conclusion that DNA is more stable than RNA. So now till now what? First step replication, in which what happened? This protein got out of the race. Why? Because protein can't self-replicate, because it do not follow this base pairing rules and all those things, whereas DNA and RNA can do so. Now, second property we will talk about stability, one of the most important property of any genetic material. But here, RNA lose from DNA. Means why? Because RNA has this reactive 2' OH group on its ribose protein, ribose sugar. Okay, whereas DNA don't have that. Even thymine, which is providing more stability to DNA, whereas this RNA has what? Uracil. So this is the reason. So now if you guys see the figure, so what the figure is showing, that it is showing normal things, that this is the ribose, okay, and ribose has what? 2', it has OH group, whereas deoxyribose had what? It don't have any OH group, and that's why it is more stable and less reactive, whereas it is less stable and more reactive. Okay, and yeah, this figure is showing this much only. Only the one thing is that the if you talk about the nitrogenous base, so cytosine, guanine, and adenine, okay, these are common in both DNA and RNA, whereas if you talk about uracil and thymine, they are different. And we have studied that this thymine is providing more stability to the DNA. So here comes an end to the stability thing. Now the third property. Now what is the third property? Third property is mutation rate. We have studied that what should be a property of a genetic material. The property of the genetic material should be such that it shows very slow mutation, and that slow mutation also helps in what? That slow mutation should help in evolution. So if we talk about DNA and RNA, both DNA and RNA can mutate. RNA mutates faster due to its instability, because of that reactive 2' OH. It is more reactive, and it can fastly mutate because of its instability. RNA viruses evolve more quickly due to higher mutation rate. So the viruses which have RNA as its genetic material, it evolve more quickly due to higher mutation rates. And that's the reason why people are very afraid of this virus and all those things, because we know even coronavirus, you guys have seen that it has, like, every next day there was a strain, and that strain is resistant to the some medicine and all. Why? Because these viruses have RNA as a genetic material, and RNA can evolve very quickly. It has a very higher mutation rate. So here also, who wins? Here also, both RNA and DNA can mutate, but the reason is that DNA can mutate but very slowly. But RNA is very highly mutative, which is not good. So that's why here also DNA wins, and here as a point mutation has been shown that how, for example, if this um means, if even, like, for example, how should I show you? Okay, so if there is a C, one base pair, okay, and except of this T, what happened? See, this T came. So now what will happen? So because we know that C will bind to G only, okay, and now T is binding to what? A. So there will be a point mutation. There will be a change in the base pair pairing, base pair, and there will be change in this base pair, and this is what we call point mutation, when there is a change in one nucleotide position. Now, fourth, protein synthesis. We know that it should show the Mendelian inheritance, Mendelian trait it should show. So, RNA can directly code for protein synthesis, central to protein synthesis machinery, better for genetic information transmission. Whereas DNA requires RNA for protein synthesis and dependent on transcription to mRNA. So, RNA can transmit genetic material. So now, what the fourth point was that, so the fourth point that was like that it should express, like, Mendelian traits it should express. So if we talk about RNA can directly code for protein synthesis, because we know, we have studied that, like, how DNA transcribe into mRNA to translate into what? Protein. So we know that the RNA that can directly code for protein synthesis, it is central to protein synthesis machinery, and it is better for genetic information transmission. Whereas DNA is what? DNA requires RNA for protein synthesis, and it is dependent on transcription to whom? To mRNA, so that the this DNA transcribed to mRNA, and that mRNA is now going to translate into what? Protein. So RNA can transmit genetic information, and that's why, RNA use is dependent on this transcription to mRNA thing. So here we have shown what? That gene can produce protein. Transcription happens first DNA converts into RNA, and RNA converts into protein. So DNA transcribe into what? Into mRNA, and translate into what? Protein. So what was the conclusion? We have seen four properties of any genetic material should fulfill this four criteria, four properties, okay, and now we have seen each and every properties, and we have compared this DNA and RNA. So what we concluded from that DNA is a better genetic material. Why? Due to its superior stability of the DNA, it can store genetic information. RNA is better for what? For transmission. RNA transmit genetic information via what? Via mRNA and protein synthesis. So we are treating DNA as a better genetic material. Why? Because it is more stable and less reactive. It can store genetic information. It will not mutate at such a faster rate. Whereas RNA is better for transmission, because it is like doing what? It transmits genetic information via what? Via mRNA, that is the messenger RNA. Even the tRNA, which is a transfer RNA, I have told in the initially that it helps during translation process and all. Take this genetic code from mRNA and the translated amino acid, all those things, it can help in that. So now if you will see what is it is normally this, it has shown a picture of DNA and RNA, evolution of DNA to RNA, how this DNA converts to RNA and then converts, like, why that converts, because it want to transmit the genetic information and it can do it via RNA, via mRNA. So yeah, this picture is showing that. So now, what is the summary of today's class? The summary is that some viruses uses RNA as their genetic material. Genetic material must be able to replicate, remain stable, allow mutation and express what? Express traits. Both RNA and DNA can self-replicate due to its what feature? Its base pairing feature. Its rules, sorry. DNA is more stable than RNA due to its lack of 2-OH group and a presence of thymine instead of what? Instead of uracil. RNA mutates faster due to its instability, allowing RNA virus to evolve quickly. RNA can directly code for protein, while DNA requires what? DNA requires RNA as an intermediate. DNA is preferred for storing genetic material. Why? Due to its stability, whereas RNA is better for what? For genetic information transmission. So what we have studied in today's class? Like we have studied in the previous class that Hershey and Chase experiment, what they did, they have proved that it was unequivocal proof that DNA is acting as genetic material. But there are some viruses, okay, like TMV and Q2 bacteriophage. What is like they are having RNA as their genetic material. So now there should be some criteria that a genetic material should follow to a molecule should follow to become what? To become genetic material. First, it should replicate, it should self-replicate because of due to base pairing. Then it should remain stable because it has to do what? It has to store the information in genetic information. Then it should allow mutation, but it should allow mutation at a very slower rate. And why it should allow mutation at a very slower rate also? Why? Because of the evolution. Organism have to do what? Has to do go through the evolution. Now, fourth, that it should express the traits. So now, when we have compared DNA, RNA or protein, that which should like following all this criteria, protein got out in the first step only. Why? Because protein can't self-replicate itself. Whereas both DNA and RNA can do what? It can self-replicate itself due to its base pairing rule. That purines will bind to what? Pyrimidines. DNA is more second, we went to which property? We went to stability property. And what was the stability property? Says that DNA is more stable than RNA. Why? Because DNA don't have 2-OH group on its side, on its sugar, whereas RNA has what? 2-OH on its sugar. Now, and also this is the main reason that 2-OH group presence. But DNA is also very more stable because of the presence of thymine instead of what? Instead of uracil. Then the third point was that it should allow mutation. Both RNA and DNA allows mutation, but RNA mutate very fast because of the instability. Because of this presence, because it is very reactive and it is very unstable, allowing RNA virus to evolve very quickly. Then this is the third point. That's why here also DNA wins. Why? Because DNA is mutating but at a very, very slow rate. Then the fourth point was that it should express trait. But here what happens? DNA cannot code for protein directly. It required RNA as an intermediate to code for proteins. Okay. DNA has to transcribe into mRNA, that is messenger RNA, and then it will translate into what? Into protein. Whereas RNA can directly code for protein. RNA translated to protein. So therefore, DNA is preferred as what? It preferred as storing genetic material. Why? Because of its stability. Whereas RNA is better for what? It is better for genetic information transmission. And that's why we study this central dogma that DNA transcribe into RNA and RNA translate into what? It translate into protein. So it was the summary of today's class. Now first NCERT based question. So question says, what primarily makes DNA more stable than RNA? Option 1, presence of uracil. Option 2, absence of 2-OH group. Third, double helix structure. And fourth, presence of thymine. So the primarily, see here you guys see primarily what is more important thing that making DNA more stable. So we have studied it is option B, that is absence of 2-OH group. Thymine presence is also adding to the stability, but the question was asking primarily, okay, majorly. The 2-OH group in RNA makes it reactive and easily degradable. DNA lacks this group, making it chemically less reactive. This chemical stability makes DNA better suited for what? Better suited for storing genetic information. So this is the same figure that we have seen, that what is the difference between RNA and DNA, that it don't have this 2-OH group. Now the second question says that which viruses use RNA as the genetic material? Among the viruses they have given, the option they have given, which viruses among them is using RNA as its genetic material? First option is lambda-phage. Second option is T2 bacteriophage. Third option is tobacco mosaic virus. And fourth option is T4 phage. Let's see the answer. Answer is option number C, that is tobacco mosaic virus. Is an example of what? Is an example of RNA virus. The genetic material is lambda-phage, T4 phage and T2 bacteriophage is double stranded DNA. So here is the structure where you guys can see this is T2 bacteriophage, this is T4 bacteriophage, this is lambda-phage, and all three of them has double stranded DNA as its genetic material. Whereas tobacco mosaic virus is an RNA virus. For transmission of genetic information, which is better? Means they are asking for this transmission of genetic information into, translating it into protein, which is better? Is it DNA? Is it RNA? Is both of them are equally important, or it is neither? Option is RNA only. We know that RNA can code for genetic material, can code for this protein, whereas DNA needs RNA as its intermediate. RNA can directly code for protein synthesis. DNA depends on RNA for protein synthesis, and protein synthesizing machinery revolve around what? Revolve around RNA. So this is the thing that RNA first transcribe into what? Into mRNA, and this mRNA lastly translate into protein and express the trait. Next question says that which of the following is not a criteria for genetic material? We have studied criteria, like there should be some criteria that makes a molecule genetic material. So among this, which is not a criteria for genetic material? First, ability to replicate. Second, chemical stability. Third, ability to mutate. Or fourth, ability to dissolve in water. So the answer is answer number 4, ability to dissolve in water, because we know that the essential characteristics for a molecule to become a genetic material should be, they should self-replicate due to this base pairing thing. They should be stable because they have to store the genetic information. They should mutate at slower rate. Why? For the evolution thing. And fourth, it should able to express its trait. Water solubility is not the characteristic of choosing a molecule to act as a genetic material. Next question says, what did the Griffith experiment demonstrate about genetic material? Option is Is its ability to mutate, its stability to heat, its replication ability, or its coding capacity? So let's see what was the Griffith experiment that told about the genetic material. Its stability to heat. The transforming principle survived heat treatment. Okay, because we know that this he did what? He has heat killed this S strain bacteria. Still, it was able to transform this R strain bacteria. This demonstrate that what? It's demonstrated the stability of genetic material. This property was later explained by DNA's ability to do what? To renature. We know that if even like by a very high temperature, if it's denatured itself, the strand got separated. If it will get the proper condition, it will get proper everything. It will renature. And that's what Griffith experiment has shown about the genetic material. Last question says, why do RNA viruses evolve faster? We have studied that some RNA viruses evolve faster. Why it's evolve faster? Option first, they are more stable. Option second, they mutate more slowly. Option third, they are more unstable. Or option four, they are larger in size. So it has nothing to do with size, we know. They are more stable? No, they are not more stable. If they were more stable, they won't be able to evolve very faster. They mutate more slowly? No, they are evolving faster. It means they are mutating also faster. So the answer is option number C, that they are more unstable. And RNA is inherently less stable than DNA. Why? Because of this presence of 2' group and uracil. This instability leads to higher mutation rate. Shorter lifespan of RNA viruses combined with higher mutation rates lead to what? It's lead to faster evolution. Means this lifespan of this RNA viruses are short and it has higher mutation rates. That's why they evolve much faster. So what are today's keynotes? First, that DNA mutation occurs slowly than RNA mutation due to what? Due to higher stability. RNA viruses evolve faster due to what? Due to higher mutation rates. DNA self repairs, RNA doesn't. So they both, so don't confuse yourself with self replicate and self repair. Self replicate both RNA and DNA can do, but self repair mechanism only DNA has, not RNA. DNA stays in nucleus, whereas RNA moves throughout what? Through the cell. So these are the keynotes. There are some interesting questions that you guys going to answer yourself. Write down in your notebooks. Why do RNA viruses evolve faster than DNA viruses? This is something we have answered also in the today's class. The curious case of RNA. Why nature choose it as messenger but not as a primary genetic material? This is also something we have discussed in today's class, but the way of questioning is different, but the answer will remain the same. So answer yourself. Then question number 3. Explain how the 2'OH group in RNA affects its function as genetic material. So that you guys also know, because we have discussed in the class today. Everything I think we have discussed in the class. You guys just need to answer it. The way of questioning is might be different, but answers are same. Thank you. Yeah. So now what we will do, TPP question thing. Okay. So let's start today's TPP questions. Okay. So question number first says, the process of translation is associated with. So the first question is that the process of translation is associated with what? Is it with ribosome synthesis? Is it with protein synthesis? Is it with DNA synthesis? Or is it with RNA synthesis? So we know that DNA transcribe into what? Into mRNA, and mRNA translate into what? Into protein. So this translation protein is associated with what? It is associated with protein synthesis. Now Now in this question they have given the 4 options and we have to tell which of the following will be the correct gene expression pathway. So they have given 4 pathway and now we have to tell which pathway is correct for gene expression. So is it gene has this mRNA, then mRNA will this transcribe and then translation and protein, then second says that transcription will happen, then gene will come, then translation, then mRNA, then protein. Third says from gene, it will transcribe into mRNA and mRNA will translate into protein. Or fourth, that gene translate into mRNA and transcribe into what? Protein. So we have studied from the central dogma rule that gene, okay, which has this DNA, will transcribe into mRNA and the mRNA will translate into protein. So the answer will be third. Next, chemically RNA is what? Chemically RNA is dash reactive and dash stable as compared to DNA. So first question says that equally that RNA is equally reactive and equally stable as compared to DNA. Second says that RNA is less reactive and more stable as compared to DNA. Third option says that RNA is more reactive and less stable as compared to DNA. Fourth says that RNA is more reactive and equally stable as compared to DNA. So we know that the answer is option number C. That chemically, if we'll talk about, RNA is more reactive and less stable as compared to DNA. Why? The primary reason is that it has 2-OH group, okay, on its sugar. Whereas second also is that it has uracil instead of thymine, and thymine we know is adding more stability to whom? To DNA. So let's save the option. Now this question says, read the below statements about the structure of genetic material and identify the correct one. So they have given us these statements. Some statements about what? Some statement about the structure of the genetic material. And now they are saying that we have to do what? We have to identify the correct one. So let's see the each statement one by one. So the first statement says that adenine and guanine are bases that have double ring structure. Cytosine, thymine and uracil are based with a single ring structure. So we have I have shown you the structure of these purines and pyrimidines in I think first class, but you guys don't need to remember that structure, but few things you guys can remember. First thing is that adenine guanine is what? They are purines. These bases have double ring structure, okay, and cytosine, thymine and uracil, which are pyrimidines, they are based with single ring structure. They have single ring structure. So the first statement is what? The first statement is correct. Second statement says an adenine nucleotide from DNA is same as adenine nucleotide from RNA. DNA adenine pairs with uracil and RNA adenine pairs with thymine. But we know that the first of all, the adenine nucleotide and the adenine nucleotide from RNA, they are not same. How they are not same? Because we know that their sugar are different. In DNA is deoxyribose and in RNA it is ribose. So it is the first half is wrong. If we talk about second half, it is also wrong. Why it is wrong? Because in DNA, adenine pairs with thymine, not uracil. But in RNA, adenine pairs with uracil. Because why? Because thymine is in DNA and uracil is in the RNA. So, okay. Now, so second statement is wrong. So first is right, second statement is wrong. Let's see third statement. So this third statement says that the base pairing that occurs in double DNA helix, okay, and RNA is synthesized during transcription follows the rule that purine pairs with a pyrimidine. So this statement is saying that the base pairing, when this double DNA helix, the base pairing which is happening in this double DNA helix. And when RNA is synthesized during transcription, the base pairing what is happening, they are following the same rule. The rule that purines pair with pyrimidine. Yeah, it is right that even the double helix DNA helix has this base pairing that purine will pair with pyrimidine. And even when this RNA is synthesizing during transcription from DNA to transcribe into RNA, at that time also this rule is following that purine is going to pairs with pyrimidine. So option third is statement third is also right. Now see the statement fourth, that the two polynucleotide on DNA molecule run in opposite direction, so that the double helix formed has two strands that are parallel to each other. Yeah, so this is also right because we know that the both the strands, they are anti-parallel. Okay, if one is like running from 5 dash to 3 dash, other will be run from 3 dash to 5 dash. So in this statement, among these statements, what was right? First option, first statement is correct, third is correct, and fourth is correct, whereas second option is not correct. So if you will see in the option, it is option number D, at first and third and fourth is only correct. So answer is D. Let's move to the next question. So next question says that they have given us again the 4 statement and they are asking which of the following statement is false. So TMV has a double-stranded RNA. Most plant viruses are RNA viruses. Bacteriophage has double-stranded DNA molecule, and most animal viruses are DNA viruses. So most animal viruses are DNA viruses. This is correct. Bacteriophage has double-stranded DNA molecule. Okay, this is also correct. Most plant viruses are RNA viruses. That is also correct. TMV has double-stranded RNA molecule. This is incorrect. So which is false? Okay, save. Okay, save, submit. So we can see all of our answers are correct. Okay. So this is all about today's class. Thank you.